Okay, yeah. Right, it was just an extension of this. Originally, it was due on Sunday, but then available until Monday morning. So I just extended it until Tuesday morning. Same assignment. It was just extended. It was just because Monday was the holiday, then figured that rather than making the availability close on a holiday, have it close on a school day. Mhm. Okay, so other administrative things. So there's the REB is out, but it's not due this Sunday. It's due the next Sunday. So I'm working on finishing up UCEB, which will should be out, and it won't be due this Sunday. It'll be due the next Sunday. So we have Unit B planned for 2 days this week and 1 day next week, and then so these will be due after Unit B concludes. So not this Sunday, next Sunday. So that's kind of the plan there. Um, Otherwise, so this is where we left off, kind of last time we talked about how the history of behavior, we had ethology and behaviorism. Ethology was focused on the largely innate. And so thus, what is the role of, how are animals filtering their environment where their behaviors are, and we're sort of trying to decipher what's going on inside the animal by changing what's going on outside the animal, as opposed to behaviorism, which came out of psychology, and it was largely focused on the study of learning and plasticity. And so there, it was less about what the animal innately was doing, but more about what the animal could be trained to do. There's a lot of other interesting differences in the history of these two fields. So you have, it's around the mid 20th century. So you've got sort of the European school of thought is those who are mainly the ethologists. The Americans were sort of more in the psychology and the behaviorism. There was some interesting geopolitical things going on that sort of affected the adoption of either one of these and perspectives on these. There were things going on about social Darwinism and all sorts of other things like this. So there's a lot of really interesting things, if you think about the history of the time, that kind of prevented these two camps from coming together, and made other sorts of things that came up, like Ed Wilson's sociobiology, so difficult for some. And so, but ultimately it came down to nature versus nurture, and the Europeans and the ethologists were largely nature, and the Americans and the psychologists were largely nurture, and these were sort of pitted against each other as two alternative ways to think about behavior. Yeah. Well, I'm just saying, in the history of it, there were camps, schools of thought that happened to be some in Europe, some in America. This isn't important for thinking about this isn't anything I'm going to quiz you on. It's not a history course, but I'm trying to give you some context as to that there was been sort of ferocious debate about nature versus nurture over many past decades. And I think a lot of that debate was fueled not entirely by the science, but by these sort of interesting groups of camps and sort of geopolitical pressures that were keeping them from coming together. Because nowadays, we when talking about nature and nurture, it's more common for us to talk about genotype and environment. And then the big question then is, so phenotype, what is it determined by, the genes or the environment? And as we'll sort of get to here, I think the modern view is much more integrated. It just took a long time to get there. But before we get there, let's just do some quick definitions to make sure everybody's on the same page here. So when we talk about genotype, if we can go back down and say, well, let's just make sure we all know what a gene is. So a gene is this idea of a discrete unit of inheritance located at a point on a chromosome. So when we say unit of inheritance, what we're talking about there is not like inches or feet or anything like that, not that type of unit. It's a discrete thing that is passed down. It is a chunk of something that is passed down. That is, a gene. And we normally sort of think about it being as located kind of at an address on a street. And you can have multiple things at that address on a street, but that address on that street is the gene. And the things that sit on that gene are the variants of the gene, and those variants are called alleles. Allele comes from the Greek for other. So when we talk about alleles, it's all of the other types that could go in that same spot. So, so when we often use a shorthand, we could say, well, there is a gene for this. What we really mean by that is there is a variant of a gene. So there is an allele that lives at a particular place that is for this, and I'm trying to be a little cagey about what comes after the four there because it sort of anticipates some of the issues we have in mapping genotype and phenotype. But the idea there is that alleles are multiple things that can sit at a spot where the gene is, and that each one of these alleles is a sequence of nucleotides. And so it could be a very, very long sequence of nucleotides. And a key thing here is that alleles for the same gene can actually have different lengths. So that's sort of an interesting beauty of how the genetic architecture is set up, is that it's not like there's exactly 15 nucleotides and that gene is exactly 15 nucleotides and every allele has to be 15 nucleotides. That just doesn't make a lot of sense. So instead, your chromosomal architecture has all of these really interesting ways to kind of index where genes are. So when it needs to turn on a gene, it knows how to get to a particular spot and then start transcribing and translating that gene and turning it into proteins, and it knows where to stop. So one of the beauties of these alleles is they've got start codons and stop codons, and that way you can sort of have them be multiple lengths. And they just sort of have to be located in roughly the right spot where maybe the chromosome bends in a particular way, so it's close to a particular molecular mechanism, or maybe another transcription factor knows how to find it. There's all sorts of crazy stuff going on in the cells that can actually, walk down the street and find the right address. But they are sort of looking for the right address. And once you get there, the house at that address could be of different sizes. And so it's just something that I think there's confusion about that. But so all of the alleles, so every gene, you've got these different variants. And if you take any individual's collection of variants together, all of your alleles, that's your genotype. So it is the collection of gene variants that is your particular genotype. Any questions about that? It's probably all review from 181, 182, maybe a little, 340. But it's definitely stuff that the terminology can be still murky at this point. So, all right. So, and then the other half of that is the phenotype. And this is a little trickier. So the analog to the gene in the phenotype is the character. And so a character are different ways in which an organism can vary in a functionally significant way, usually observable, usually measurable. So an example of that is eye color is a character. I can have a different eye color than you can have a different eye color than me, and all over the room. That is a character. So that sort of stores the different eye colors that we can have. Now, the particular eye color is what we refer to as a trait. So, I might have brown eyes, that is my trait. And you might have blue eyes and so on and so forth. And so that's kind of like allele and gene, trait and character. Now, the combination of all of those traits together is your phenotype, collection of all of your traits. And so there is some, nice parallelism between these two, but we don't want to make the mistake of assuming that there is an obvious connection of every gene to every character, every allele to every trait. And that's part of what we're going to get into in this section, is that there's genes and they're important for inheritance, But phenotype is a much, much more complicated beast. And the type of one-to-one gene to phenotype mapping that maybe you've heard sort of in introductory biology doesn't really map onto a lot of the traits that we often see. So it's you kind of have to think of these somehow as separate. They're certainly related, but as we'll see, the phenotype is the result of the expression of potentially very, very many genes. And so it's often inappropriate to say that you have a gene for brown eyes, for example, or even an allele for brown eyes. You just have a brown-eyed trait. That's your trait, brown eyes. All right, and we'll get into that a little bit more. But questions about these basic, these terms. Everybody on board? Gene, allele, genotype, character trait, phenotype. Okay. And then the connection between these two really is the nature versus nurture debate. So, if you think that all the phenotype is just directly determined by the genotype, you're on the nature side. If you think that the phenotype is plastic and can be molded by the environment, then you're on the nurture side. But I think the modern way to look at this is there's no side. So this, is kind of the old way of saying it, nature versus nurture. Is it the G or is it the E? But some kind of more modern way of thinking about it is not versus, but via. So nature via nurture, or the way, biologists like to say it is G by E. So that's what this GXE is. We just pronounce it G by E. And sometimes we say G by E to P. We're just shortening it by G by E. So gene by environment interaction. So that is really the way we view phenotype now, is that the phenotype is a product of these two things, the genetics in an organism plus the environment. And it can get even a little more complicated because there's this thing called epistasis or epistatic interactions, where it's the gene in the environment of other genes and the environment. So this is when you have, and I'll give an example of this in a second here, but when you have a gene that its interpretation varies depending on what other alleles are there, then you have this epistatic interaction. And this is sometimes people shorten it and kind of be cute here by saying G by E is sort of our approximation of that. So this is kind of the way we think about how we get phenotype. And an example of this epistatic interaction, so kind of grouped this G and E together. And this is just kind of a common cartoon example, but let's say you had an allele for blonde hair, that trait, let's say it's a very simple mapping of traits, or you have an allele for red hair. And so then there's also an additional allele for baldness. Now, if you don't have the bald gene, the bald allele, then you have hair color, blonde or red. But if you do have the bald allele, the bald hair gene here, then it doesn't matter what gene you have for your hair color because you're just bald. So that's an example of an epistatic interaction where you technically have the gene for blonde hair, but you don't have hair. And so you're not going to see the expression of that gene. So the interpretation of having that gene only makes sense in the context of another gene. So epistatic interactions, epistasis. And that goes into the complications of trying to map one gene to one character, one allele to one trait, is that there's this stuff shows up all the time. And then there's other things too, like environmental constraints. What if, in order for me to make brown eyes, I need a pigment that I get out of my diet, and that pigment is not in my environment? Well, I have an allele for brown eyes, but I can't have the brown eye trait, because in order to have the brown eye trait, I need a nutrient from the environment that I can't find. So what color are my eyes? So that's another example of how the expression of these genes is going to be conditioned on all sorts of things, why it's really kind of inappropriate to sort of say that we have genes for traits, when in reality, we just have interactions of genes in the environment, and it just gets kind of complex. Okay. So questions about epistasis? Hopefully this is also a little bit of review. Okay. All right, so G by E. So really the question is not which is more, is it G or is it E? Is it nature or nurture? But it's the question is what is the relative strength of the G or the E? So if I have a particular phenotype, is it mostly determined by G, mostly determined by E, or some balance between the two of them? That's kind of our modern perspective. And often then what we do in animal behavior is try to come up with clever experiments that tease these two apart. And in this unit, we will go through some of the different methodologies for teasing the two of these apart so we can test hypotheses about causation. Let's say mechanistic causation. So, is this due to the presence of a gene or is this due to something in the environment, for example, or evolutionary? So, if it is something that evolved over time, then perhaps we would expect to see certain signatures in the genome that may not be there if it is just a product of the environment, and so on and so forth. And so we hopefully can build tools that help us figure that stuff out. So one of the things I'm talking about strength here is a term that we should be familiar with is heritability. And so this is a tricky term. It is not inheritance. It is not about individuals. This is a property of populations. So you can't assess the heritability without looking across a population. So heritability is the proportion of phenotypic variation in a population explained by genotypic variation. So basically, if I look over a population, I can see how much variation do I have in hair color or eye color. And then over that same population, I can say how much variation do I have in alleles. So what is the distribution of different alleles out there? And then I can ask, how does variation in the genes, in those allele frequencies, predict variation in the hair color? And if there is a strong match, so you can if you're far off, like if you have a weird allele and you also have a weird hair color, or you have a common allele and now you have a common hair color, then in that case, we would say that hair color is a highly heritable trait. But if it goes the other way around and there's a lot of genetic variation and there's maybe a lot of phenotypic variation, but there's no common patterns, that's not heritable. Or if there's just no variation, so if you purge the genetic variation, so then you have phenotypic variation but no genetic variation, that also gets rid of the heritability in the population. So, and so we'll see a little bit more of examples of that. But so heritability is a function of the population. We often use this uppercase H squared for it. And there is this idea of broad sense and narrow sense heritability. If you want to get into the weeds, has to do with sort of what type of genetic variation that you're accounting for. I, at this point for this lecture, am not don't want to go that far. It's a little out of scope, but it is it's actually covered in the Breed book a little bit, and we might talk a lot about it a little bit more. But for now, just look for whatever the case, H squared. And that squared, where's the squared come from? It's a little bit like the R squared and a correlation coefficient. If you're looking at data, if you're looking like a regression line, it's strongly related to that. And we'll see some examples of that when we look at some data in I think either Thursday or probably Thursday when we look at that. So heritability measures this relative strength of these two things together. And if you want to think of it rather than, so when I'm using this delta, this is what I'm using for like the variation in phenotype due to variation in genotype. So this is like, I really probably could have done like delta little gp or something like that. Then it get a little bit ugly. So all I'm trying to say is that how much variation is explained by variation in the genes. And so this is sort of the bigger picture of heritability here, is this is saying that how much of the phenotype is explained by the variation, how much of the variation phenotype explained by variation in genotype, divided by the total variation in phenotype, which we assume has got two parts: variation due to the genotype variation and variation due to environmental variation. So if the phenotypic variation is all environmental, so like I ate a carrot, so I got orange hair. You ate a plum, so you got purple hair. In that case, there would be no heritability. It's all about what you eat. But if on alternatively, if there's a lot of environmental variation, but that is not coming through, so that my genes seem to be a strong predictor of my hair color, then hair color is heritable, and we can do this for behaviors as well. All right. So, any questions about heritability? Yeah. Well, that's sort of heritability is meant to capture the relative strength of the genes over the environment. So, yeah. Um, and it's a tricky thing because it's population. There's like people will talk about, for example, in dogs. There's a paper recently written about the genomic architecture of dogs and how that relates to their hypersociality. So it was a behavior paper, it was a behavioral genomics paper. Maybe it was a good one I could put up on the as an auxiliary reading or something like that. But they pointed out in it that of dog breeds, sociality is like highly heritable in golden retrievers, but is not heritable in huskies, I think. Let's call it huskies and golden retrievers. And that is a very confusing statement to someone who's not a specialist. That might sound like they're saying, well, what do you think that sounds like it's saying? It's saying, again, hypersociality. So how whether a dog will socialize with a human. They're saying that's highly heritable in golden retrievers, but not in huskies. What are different ways you can interpret that? Yeah. Well, I would think that so your that response, and thank you for that, reflects one particular interpretation of that phrase. Hypersociality is highly heritable in golden retrievers, and not so much in huskies. And so, if I was to take from that away from it, then it feels to me that your response to that statement is that golden retrievers are more social to humans, and huskies are more social, and that might be the case. But is that what that statement is saying? Yeah. So that's because that still to me that feels like a very individual focused so as opposed to being a population focus that seems like it is still attributing an attribute to the breed, and right now the only attribute we're attributing to the breed is the heritability. We're not actually saying anything about the sociability about the we're saying that hypersociality is highly heritable in golden retrievers. We're not saying golden retrievers are highly social. Did you have a comment? Okay. Well, I like this shift to breeding. So let me go back to what you were saying. If you were choosing a dog to breed for being social, for training purposes, not this dog, but this dog's progeny. So if you wanted to produce a line of highly social dogs, would you start with a golden retriever or would you start with a husky? And why would you say that? Mm, okay. Mhm. Okay. I mean, that's still to me that is very interesting, and but yeah, it feels to me that it's still more a comment about the individual. I saw your hand up first. Okay. I like that comment. I'll repeat that in a second. Did you have another? Uh-huh. That's a good question. Does heritability does that mean there's more variance in behavior? And I think we're getting. I like the answer in the back here, where it was about if you were to choose who to breed. What heritability is telling you is that if you look at the variation across the genes, so let's look at all the huskies. Let's genotype the huskies and characterize how different their genes are from each other, and let's genotype the golden retrievers, and let's see how different their genes are from each other. And now let's phenotype. Let's actually assess each husky's how social they are, and then let's assess each golden retriever how social they are. What high heritability means is that there will be a strong relationship between the genetic variance and the behavioral variance. So that would mean that a highly social golden retriever will have a gene variant that is very often in golden retrievers associated with being highly social. And likewise, a less social golden retriever will have a gene variant that, for most golden retrievers that have a gene variant, are also not very social. But if you go over to the husky, then there's just no relationship. So there could be highly social huskies. I'm not saying there are, but there could be highly social huskies, and they're going to have genetic variants as well. But that sociability of that husky is not predicted from their genes. So if you went and genotyped that husky and sent it off, then it would say it's got these gene variants. And that wouldn't tell you anything about their sociality. So for that gene variant, your husky could be social or might not be social. It really depends on the environment that they were grown up in. But for your golden retriever, if it's got the less social variant, it's probably not social. And if it's got the more social variant, it's probably social. So getting back to the breeding question, a reason why you would pick if you were breeding for sociability, you'd like to pick a subset of a population for which ability is highly heritable, because then that would allow you to pick out a highly social set in this generation, breed them, and you would be then very likely to then, because the gene is the unit of inheritance, because there is often a relationship between the genotype and the phenotype in the heritable case, then you'd be very likely to get a set of offspring that are going to have the heritable sociability, and then you can keep selecting and selecting and selecting, and then that allows you to then generate this highly social line. Whereas if you tried to do it with a husky, if you pick a really social husky, the low heritability of sociability in the huskies means that its offspring are just as likely to be not social as they are to be social. So that's what heritability is. It's what is the relationship in a population between different genes and different behaviors. It is a property of the population, not of the individual or the breed. And that was one of the things that in that statement that they wrote in this introduction, that this the golden retrievers have a highly heritable hypersociality, but huskies do not. Then the behavioral genomicists here were criticizing that, because that's a really that is a difficult thing to say. People are going to read that and they're going to think that they're saying golden retrievers are highly social and huskies aren't. And they're not saying that. They're just not commenting at all about the variation in sociability in these two breeds. They're just saying, if you look across the breeds, the variation is explained by genes in one, and it's not explained by genes in the other. That's heritability. It's environment with huskies, it's genes with golden retrievers. That makes sense. So there are questions about that. And that's what I mean by strength. The genes are very strong for sociability in golden retrievers. They're very weak for sociability when it comes to huskies. And huskies, the environment's stronger, and golden retrievers, the genes is stronger. And that comes out by saying that the heritability is stronger for this trait in golden retrievers. That makes sense. It's a complicated idea, especially because it feels like it should be an individual thing, when it really is a statistical property of populations. And we'll do some heritability calculations in probably again Thursday, where we'll see how this is kind of calculated by plotting these sort of things out. And you see it's just kind of a statistical regression, which again goes back to why this looks like R squared, which is, how much variance is explained in a regression line. Okay. All right, great. So that's the term we use for the relative strength of G. Questions? Okay, I think we covered that. Let's then do a little attendance thing. I'm going to try, supposedly I can do this from my phone. So multiple choice, play. I'm skeptical, but we'll see if it actually starts. If it doesn't, I guess I'm just back here anyway. Oh, right, I need to do the first time from here. Okay. All right, so it should be started. So what term represents the collection of all an individual's traits? So far we've seen a term that represents the collection of all of an individual's alleles and the collection of all of an individual's traits. So looking for the trait one. And I think we have almost everybody in. I think most everybody in. So this is kind of bimodal right now. Right now we've got 3% for heritability. We've got 81% for phenotype. We've got 6% for genotype and 10% for characteristic. And I totally get that, the characteristics, because that traits are characters, and so characteristics is kind of the simple word we have for it. But the jargon that we use, which is loaded and captures everything that I'm looking for here, is phenotype. And that the reason we use phenotype and not characteristic is that it reflects all of this delicate balance of gene and environment interaction. So that's the reason why we have the jargon. When in the end of the day, what is a phenotype? A phenotype is just a set of characteristics. But you might also think characteristics kind of sounds like a set of characters. So, we're really looking for the traits. And so that's why phenotype is the term that I'm looking for here. All right. Stop the poll. Okay, so relative strength, modern perspective. Okay, so if we think about the modern synthesis here, so it's been over time, there have been a number of different methodologies that have kind of folded into what we now think of as the mapping from genes and environment to phenotype, the just G by E. And I just want to walk through a couple of these that I think are kind of interesting, or some interesting examples from them. And so, domestication, for example, Darwin was extremely motivated by looking at, well, a number of cases of domestication, but in this case, these are Darwin's the pigeons, right? So so he was interested in the ways in which humans were artificially selecting for traits. And this is one of the things that started Darwin thinking about, on the origin of species, about how you get these traits spreading. And when we think about history, and I've already talked about the gene being, and we'll get to this when we talk about quantitative genetics, but the gene, Mendel, there were the Mendelians and the Darwinists, and they really disagreed about inheritance. I mean, Darwin looked at the ways in which you could select for different traits and saw continuous variation everywhere. And I'll get to your question in just a second. And that's what led him to think about natural selection. And if you think about the description of natural selection, it is all about continuous variation. Like, it's, at least that's the main conceptual model. And so Darwin had a hard time thinking about a unit of inheritance that was discrete. How could you inherit a discrete chunk of whatever inheritance is and somehow get a continuous trait out? That was like, that didn't match his experience. And a lot of his experience came from looking at the ways in which humans were doing effectively artificial selection. Question. That's Darwin right there. Yeah. Charles Darwin, the On the Origin of Species guy. That is Charles Darwin. Yes. Mm-hmm. So, yeah, so domestication was, a very early influence. But then, when phylogeny was kind of became, when Darwin's sort of idea, gave strength to the idea that, that things came from other things, and this was a backward, directed tree. And so, phylogeny became kind of a way to capture that. And we already talked about chronograms, so a particular type of phylogeny which captures the time since things have been divided. So this is an example of something simpler, a cladogram, and so, or cladogram, I guess. And so the idea here is that it's similar to that that chronogram that I showed, where it had all of the straight lines, where the length of the straight lines was time. But in this case, we don't care about time. We're just talking about evolutionary relationships. So in this particular case, we've got the evolutionary relationship between these nesting birds, and we see there's several different species here. And so they took molecular or morphological differences between these birds and then came up with this. So you can imagine, before you had molecular techniques, you could just say, well, these birds share a bunch of traits, but they differ by a little bit. So, like, birds of this type never have quite the same beaks as birds of this, but everything else looks about the same. Well, you imagine that they should kind of cluster together. And birds that look totally different should cluster away from that. And so you could put all those traits into a giant bag and then use special methods that group them together in this thing called hierarchical clustering. And then that ends up forming these cladograms that sort of show that, like, well, these two species are more closely related than, at least in terms of their morphology, than, say, this species and this species down here. It's how you might read that. And you can do that again with observable morphological traits, or if you look at their genes, you can then ask how similar are the genes of these different species. So if you look at all the genotypes of the different, these birds that we collected and all of these, then we could toss those all in and then cluster them by that. Now, what's the cool thing you can do about these cladograms is if you take the morphology or the molecular cladogram for granted is the right evolution, then you can map onto that behaviors. And so in this case, these are different ways in which these birds build their nest. And so this one here, support, which is either orange or white, I think if it's white, that means they actually form a glue, that they glue their nest to the side of a rock surface, versus I think the other color, they find an edge and they just set their nest on top of it. And you might imagine that if, say, the behavior of gluing their nest to the side of a rock surface was sort of, it evolved, if there's a common ancestor that had that, then you might expect to see it across all of these edges here. But what we end up seeing, and I think again, orange maybe is glue and white is just find a rock surface, then you see that there's white everywhere, but orange only shows up here. And so when we see that this behavior, like the more common behavior, we might then say a more parsimonious explanation is that whatever this white behavior is, let's say it's the rock surface one, that's probably the ancestral state. And later in evolutionary history, that's when the gluing behavior showed up. And this is really important because, as we will talk about in this unit, finding the genetic basis of behavior is really, really difficult. Finding the genetic basis of, a compound that you produce on your skin is a little easier, but finding it for a complex behavior, like whether you glue your nest to the side of a mountain, you might not actually have any molecular evidence that could help you figure out which came first, the rock face or the glue. But if you have other evidence that sets up this cladogram, then you can use the observations of the behavior in the cladogram you take for granted, and then from there test hypotheses about what's the most likely evolutionary trajectory for this behavior. And again, the orange behavior probably showed up later, and the white behavior probably was the one that was in common. It's probably that, because the alternative is the orange behavior was in common, and then it got lost all of these times. And that might have happened, but it just seems like the more parsimonious suggestion would be that these two came from a common ancestor that evolved that behavior, and the rest of them were stuck with the ancestral. So that's kind of how we would use the phylogeny to inform how we think about how behaviors build upon other behaviors. So we talked about phylogeny. Question, yeah. I'd say for the animal behavior course, I think you should know how to interpret the phylogeny and then apply it as a tool for understanding the evolution of behavior. So it'll be less about the the actual building the phylogenies, but more about if I give you a phylogeny and then I ask you questions about how the behavior relates, then how can you use that to talk about the behavior? Yeah. Yeah, right. Well, I mean, I mean, names are sort of, I mean, that's just biology. Unfortunately, they, have these kind of, I can try to make the names. If I try to make it more real, then it's going to look more like this. If I try to make it more fake, then it's going to be A, B, C, D, and it might be hard to sort of, well, what is an A bird versus a B bird? Yeah. This one here, right? Well, I mean, this is the best resolution that they could get with the data that they had here, and so it's they're saying sort of these three have this kind of common ancestor, and so there's not necessarily an ordering they can get with the characters that were available to them. Yeah, I mean, I remember it's really this is about hierarchical clustering, so it's about like the closeness among the leaves, and so it's easier to sort of say like this one is closer to this one, or like that it's more about comparing pairs versus other pairs, and then that hierarchy that forms from those comparisons helps us make assumptions about where things came from. But again, I more just want you to be aware that evolutionary histories can be constructed and that they can be useful for talking about where behaviors come from. And without things like this, then sometimes it's just so stories. Because the thing that you might think is the ancestral variant might actually be the thing that crazily enough was lost a bunch of times or whatever. And so the being able to reconstruct these and then intersect them with behavioral data gives us a lot more richness in understanding what behaviors came from what other behaviors. Okay. All right. So phylogeny and then quantitative genetics. So this is this is the thing that really bugged Darwinists about Mendelians is that variation you observe in nature is usually continuous. So these are so-called quantitative traits. So, that whether a leaf is green or, whether a leaf is smooth or has ridges, that's a discrete trait. So when we're talking about bean plants, there's a lot of these kind of discrete traits, but a lot of the traits that we experience, height, weight, those sorts of things, if you look across a population, you don't just have smooth leaves and rigid leaves with ripples or something like that. You've got an average height and then variation around that height. And so that's what we refer to as a quantitative trait. And so the difficulty when Mendelian genetics came up is saying, well, if all inheritance is based on discrete units, so you're being handed down a discrete chunk from your parents, how could you possibly get a continuous trait out of that? And so for a while in the early 20th century, people had to try to figure out conceptual models that would produce these curves, these distributions from inheritance structures that seemed like they must be discrete. And for a while, Darwin and people who came after Darwin refused to adopt these Mendelian gene ideas, because they came up with these other ideas of like, well, maybe there's this sort of like soft stuff that you can get more of and less of, and when you inherit it from your parents, then they can give you a little less of it or a little more of it, and that's what gives you sort of this variation there. But then as the molecular methods got better, then eventually all that kind of got put to bed, and people said, no, we are going to have to make sense of this. There has to be a way to go from discrete inheritance to continuous traits, otherwise known as quantitative traits. And the ways in which we think about that now goes back to G by E ideas. And so there are a bunch of variances here. So we don't now think that every trait is tied to a single gene. Every trait's tied to a bunch of genes. So if every trait's tied to a bunch of genes, like if there was a gene and had two alleles, I could either have one allele or have the other allele. I could either have ears that were connected or ears that were separated or something like that. But if I have multiple genes, then and each one of them has multiple alleles, then now the combination of those genes gives me a lot more to work with. So I go from having 2 or 3 different trait values to having 3 times 3 times 3 times 2 times 4 or something like that. So now the portfolio of those alleles helps to determine this smoother variation. You take all of that gene stuff And then you add it to variance from environmental effects. I mentioned to you, if my eye color depends on me finding a carotenoid or something like that, and finding something in the environment in order for me to synthesize that eye color, if I find less of it or more of it, that is going to create a variation in my eye color. So I will have a genetic predisposition to a particular eye color, but then my environment will perturb it one way or another. So the genes and the environment together give you plenty of ways to get quantitative traits, to get continuous traits out. But it just hard to then figure out to disentangle, for height, what is the gene for height? Well, I just said there can't be a gene for height. Height is so continuous here. There have to be multiple genes. And so when I'm thinking about quantitative traits, I need to be thinking differently about where to look for them in the genome. And that's something we're going to talk about a little bit, the methods for that here, I think, on Thursday. So that'll be the QTL, the quantitative trait loci, the places sort of on your genome where one trait happens to have representation. It's not one place, it's many places acting together. So that's what I mean by quantitative traits. Does this make sense to me? What does sense everyone mean by quantitative traits as opposed to discrete traits? And how the existence of quantitative traits kind of tells us that the mapping from G to P is very complicated. It involves lots of G's potentially and a bunch of E's. And if the variation, that's why we talk about heritability, is that, in heritability, some of the phenotype is coming from the environment, not the genes. So already you've got, this stress that, things moving things around. All right. So, and then evolutionary population genetics, favorite topic of mine. We talked about this from last time, these sort of 4 different forces. And I just want to highlight here, we talked about these 2 reduce diversity in populations and these 2 increase diversity in populations. And something that you might think about there, now that we've introduced heritability, then we can start thinking about how evolution affects heritability. So if you're reducing genetic variation in a population, you're actually reducing heritability in that population as well. Because before selection, then you would have found that there's a lot of trait variance and a lot of gene variance. And if they were highly heritable, then the traits that were at a disadvantage will gradually start disappearing from the population until you're left with just the genes that code for traits that aren't kind of getting in the way. But at that point, you're still going to have trait variance, but it's pretty much going to be due to all environments. So that's kind of an interesting process, is that heritability gets purged from a population by selection itself, where selection, though, only works with heritability. It's kind of beautiful. It's a regulating loop where you need a heritable trait to be selected on. And once selection acts on it, you get rid of heritability, and thus it turns off selection because selection can't act on it anymore. Yeah. Pardon me? Purge means removed from the population. So if you have a bunch of different alleles, and these ones are much better at reproducing than these ones, then these ones selection will get rid of, and all you'll be left with is the ones that reproduce very well. You're still going to have variation, though, but all that variation will be environmental, so it won't be heritable variation. Heritability is removed. So selection removes genetic variance. Genetic variance removes heritability. So heritability is a dynamic process changing over time. And if something's highly heritable, then that's why breeders pick it. That's why, I asked, do you want to breed sociability into a golden retriever or a husky? You breed it into the heritable golden retriever. But eventually, once you get the most social golden retrievers as possible, there's no way to make them more social. At that point, all the variation in their sociability is not coming from their genes, it's coming from how they were brought up, their environment, their diet, etc. Okay. Any other questions about that? All right. This brings up an opportunity for me to talk about one more term that you should know, ecotype. So if you have a species and it gets separated from its, if you have a population and they get separated and they're put into two different habitats over time, then you're either going to have selection or drift. So usually when we talk about ecotype, it's just selection that's going to tailor that subpopulation for that environment. They still can mate with the other, the group. So there could be, if they ever came back together, we would still view them as one species, but they've now kind of been specialized. And so we refer to them as ecotypes, genetically differentiated populations that are environmentally selected for a particular habitat. You might have heard of Africanized bees. Africanized bees, which are the only bees, the wild bees that we have here in Arizona, are bees that got a very special aggressive phenotype in Africa, likely due to also, natural pressures there. That bee was then bred into a European style, more docile bee for the hopes that it would make it a more robust bee, but it ended up giving it so much of an advantage that when it got out, it sort of took over in areas that were warm enough. And so, once you get, too far north in the United States, you lose out on Africanized bees because it's too cold for them there. But down here, it's plenty warm for them, and they can outcompete all the other bees, and they can mate with the other bees. In fact, there's weird environmental things that can happen where if you get an Africanized bee in a colony of a European honeybee, that actually will lead to Africanized effects in the behavior of the existing bees. So there's a bunch of weird stuff that goes on there, but that's an example of an ecotype. And so it is still Apis mellifera, it's still a honeybee, but there are a variety of ecotypes of honeybees that have their own little traits that are tailored for the environment that they come from. The Italian honeybee, the European honeybee, the African honeybee, and so on. So ecotype. Is that clear? Okay, great. All right, now the fun stuff. These are just 4 pressures that I'm glad that Breed brought up because I think they're pretty cool too. And it gives me an opportunity in this boring lecture so far to show a little bit more animal examples here. When we talk about evolution, evolutionary pressures on behavior, one of the things we need to talk about is weird ways in which behaviors can hang on that aren't quite due to drift exactly. We talked about genetic drift before. They're due to these other pressures that cause them to be maintained in a population, and you wouldn't otherwise kind of explain that or expect them to be maintained. And one of them is correlated characteristics. And so this is a trait that, let's say, is important in one sex, but is produced by both, likely due to, say, a developmental constraint. And so it ends up being too costly to get rid of in one sex to warrant getting rid of it in the other sex. Or in other words, it's too beneficial to one sex and that outweighs the cost of it in the other sex. So there are simple examples of this. Nipples on male mammals, for example. They have no function from a mammal's point of view, but getting rid of them on all of the males in a mammal population would require such a costly change to development that it's not worth it. And getting rid of them at all, well, mammals, the key defining feature of mammals is they drink milk. So you probably don't want to take away the nipples on a mammal. So you get them on men. Likewise, horns and antlers and ungulates that are used for male-to-male competition. So that's an example of a trait that is only used on the male side for this male-to-male competition, and yet you still will get the expression of these traits in females. And then mate choice. And these are a bunch of great examples in mate choice. This is a classic one in these barn swallows. And so if you've ever looked at a barn swallow, they have this tail, sometimes known as a swallowtail, right? So it has these very long feathers on the end of it here. And it turns out that females prefer male swallows with very long tails. And so that sexual selection, which we'll talk about later in the semester, is one of the things that drives male tails to get longer. But interestingly enough, that leads to females with longer tails. Now, they've shown that females and longer tails do not get any more attention from males. So that does not seem to provide them any benefit. In fact, it actually provides them less offspring in the long run. So they've shown that the difficulty, the added difficulty from flying due to these long tails ends up making the females worse off in the long run. So you can, and I'll show you a study in a second, you can artificially extend the tails of swallows. This is the type of stuff you do in animal behavior. You can tape on feathers to the end of a swallow and then let her go on with much longer tail, and then watch what happens to her reproductive success relative to others who haven't had that treatment. And what you do find is that they have less eggs in the long run, but it may be that they compensate by actually putting more parental investment into the eggs that they have. So it's this interesting thing where you're stuck with this trait that, because the females prefer the males with the long tails, that leads to females keeping their own long tails, which puts a cost on the females, which seems to be compensated by additional parental investment in the fewer eggs that they lay, because they can't, they have this performance hit. And if you want to read more about this, there's, I've linked to this, and that this is an example where they did experimentally elongate the external tail feathers of females, and then showed that they do have less offspring, and that potentially seems like they have more parental investment. I mean, you can. This is meant to be a primary source backing up what I'm saying here. So if you're interested in reading more about this behavior, then you can feel free. I'm not going to quiz you on this particular paper, but I do want you to know about these correlated traits. So the correlated characteristics, that's the thing that I'm most interested in. I'm just trying to give you real examples of this, and that's why I put up this paper. So correlated characteristics, swallowtails, cool example of that, I think. Phylogenetic inertia. This is my dog, Dexter. This was him as a puppy. This is that office chair. This is that same office chair. This is him a little bit grown up. He is in this position and in this position because ever since he was a puppy, he liked to spin before he laid down. And then when he gets done spinning, he plops down into this very, very tight ball. And that's just something he does. And if you've had a dog, the answer is dogs do that. So people have asked this question, why do dogs do that? And the best explanation that people have is it's something called phylogenetic inertia. So basically, these are present day ancestral traits that are no longer adaptive, but aren't also very costly. So you just have them. So the thought is that once upon a time, there were wolves or foxes that needed, before they laid down, to survey the area for, ticks or sticks or, there's a number of different things that they, was apparently adaptive to do before you go and lay yourself down for hours on end. And dogs don't have to do that anymore, but it doesn't really hurt them to do that. And so it's a behavior that's just going to be stuck there because it's probably too costly to rewire them than it is to just keep this relatively benign trait. So that's what we mean by phylogenetic inertia. It's just baggage of a behavior that doesn't make sense anymore, but we still have it. That makes sense? The other cool one here, disruptive selection. So this is a fun example. So the idea behind disruptive selection is that you get two phenotypes, two traits in the population that are maintained despite being very different from each other. And this is common in sort of there's these kind of sex examples. So this is in cuttlefish, this female impersonation example here. So and I just put this up here so you can read about it, but basically what it's saying here in the highlighted portions is in South Australia, they found a giant aggregation of these cuttlefish, which are these cephalopods, and there was a bunch of males and a bunch of females. And most of the males have this phenotype where they get into fights with each other and they aggress in order to win access to a female. Now, cuttlefish, the females display a different look than the males. And so I can kind of show these things. So like you can see that there's a lot of variation in the way these cuttlefish look here. And there are some consistent, so the cuttlefish can actually change the color and texture of its basically of its skin. And so the females tend to look different than the males. And so what they documented in this case here is that while there are males that are fighting, like I think there are two males here that are fighting for access to a female, or maybe it's over here, there is a third male that puts on a show and looks exactly like a female, slides in behind the two males that are fighting. And then while the two males are fighting over here, then it turns on its maleness and starts displaying for the female that it's just got access to, and she mates with it. So that's an example where there are these two phenotypes. There's one where it's kind of this aggressive male, where they get into fights with other males in order to win access to a female. And then this totally different trait where they slide in and they actually are cryptic females where they can kind of come in, or cryptic males, I guess, so they can pretend to be female to get access to the female. So those are two strategies that apparently are both being maintained in this population, either hyper male or kind of sneaky male. Yeah. Well, that's the idea. There are some males that can temporarily look like a female, and that allows them to not get into fights with other males. So they can sneak around the other males that otherwise are guarding. It's a terrible term, but they're guarding the female, and then by sneaking around the guards, they get access to the female to do their display and mate. Whereas any other male that doesn't have this trait would actually get into fights with the guard. So that's disruptive. And then the last one I wanted to kind of cover here, the handicap principle, which we've seen before. The stotting behavior I talked about in lecture 0, where it is this prey is so fast that by jumping up in the air, it still has enough speed to outrun a predator. And that is so conspicuous that predators that have learned to not chase that prey down do better than predators that don't learn that. And that because this handicap is an honest signal that, if this prey is still around, then that must mean that they're so fast that it's worth just paying attention to the signal and going after another prey. That's one example of handicap principle. If you just look at that right now, you'd say, why would that behavior ever stick around? It's weirdly because it's a handicap. Another nice example of that is the peacock. And so, there's this beautiful mating display, and you can talk about like all of the interesting signaling that goes on between the male peacock and the female peahen, and fine and all of that. But still, how the heck did this thing evolve? Especially because if you look at the male peacock as it's flying, this is clearly a huge handicap. And it also is very conspicuous to predators. So Zahavi, who's a sort of famous evolutionary biologist, he's sort of the one that's kind of associated with this term handicap principle. And his explanation is that it's actually the fact that you can find a male that can display this shows you, honestly, as a female, that this male must have other interesting things going on, because otherwise it'd be dead. So there's no way you could survive with this handicap unless you also have access to all sorts of resources, you're tough, you can stay out of harm's way, all of these things that might be good other traits. Yeah. Well, it makes it difficult for it to fly, but it doesn't have advantages in its own. And that's the beauty of it, is that because it's such a handicap, the fact that it still is alive, despite having that handicap, means that there are other things that must be good about this peacock that the peahen should be willing to mate with. That is the handicap principle when applied to sexual selection. So before Zahavi, the statistician Fisher thought that these sorts of traits, these beautiful tails here, were arbitrary characteristics that are just kind of a product runaway. Like, oh, I kind of like a red spot, and because I like a red spot, then my daughters will like a red spot, and so on and so on. And then the red spot gets redder and redder and redder, and it has no fitness value whatsoever. Zahavi was the first one to kind of come up with this idea that actually it communicates fitness because the rest of the bird must be pretty darn fit if it can manage to sport such a handicap. That's the handicap principle. And that's why these crazy behaviors and other kind of morphologies potentially could stick around. Okay. Any questions about that, the handicap principle? All right. All right. And then there's molecular genetics, which I'm not going to talk about too much here. You know, it maybe I'll talk a little bit at the beginning of the next one about kind of the ups and the downs of molecular genetics. But the key questions that we worry about, how to discriminate and how to quantify. So that's what we'll talk about for the rest of the unit. Reminders about the perusal stuff. And then let's do this one last eye clicker and get you guys out of here. So I'll do a short answer. And the short answer here is what is the name of what do we call a subpopulation that has been separated from its main population and then has had genetic adaptations that specialize for its new local habitat? So what are sort of subgroups of species that are genetically specialized to their local habitat? What is the term we use for that? And that's all I've got for you. Yeah, the question was, what is the what do we call, what is the term for subgroups of populations that have become subgroups of species that have become genetically tailored to their local habitat? Well, put in put in the answer that you think, and they're not graded for correction; they're graded for completeness. So I just want you to think about it, and then you're feel free to look through the chapter to find the right answer. I would look through the notes and the chapter, and then you can figure that out. Pardon me? That's fine. It's graded for completion, not correctness. That's right.